Hello and welcome to BioCode. In this video, we will see how we can utilize alphabet class from BioPython that allows us to understand the alphabet that underlie within the sequences of our analysis of whatever sequences we are utilizing. Basically, it allows us to understand the string. For example, if you are inputting a particular file, if you are giving a sequence directly to the on the console of the Python language. For example, if the sequence looks like this and consider it is a protein sequence, so obviously there is no J in the protein, but consider it is a protein sequence. So how will Python understand that it is a protein sequence? Because there are quite a lot of possibilities that a protein sequence can contain up to 20 amino acids in itself, a multiple of 20 amino acids. So how can it understand that it is either it is a RNA, either it is a DNA, either there is an ambiguity within the sequence, if it is a real sequence, if it does not have any issues. For example, sometimes DNA sequences or nucleotide sequences do have repetitions such as n in capital or small letters, and amino acids can also a protein sequence can also have amino acids that are designated as X, which are usually considered to be repetitive or unknown amino acids. So to understand these sort of issues within the underlying sequences that we input as to the Python language, to understand those sequences, Python has to utilize some of the BioPython functionality that BioPython allows us to figure these sort of sequences. And basically, if in previous videos, as you know, we try to input the sequence and then we convert it to a sequence object. uh, Basically, that was not the actual way to create a sequence object. We obviously did create a sequence object, but that is not how it is done in the industry. It is not how it is done in the professional life. So to be able to create a sequence object, we also need to provide its alphabet. It is basically an optional, but as I said earlier, that it is professionally sane to actually utilize the alphabet of of the sequence. If you already know it, you can input that a particular alphabet yourself. So basically, what is alphabet? Let's discuss that. uh, if you know IUPAC, you can if you don't know, you can read our article about that. And if you know about IUPAC, basically it is an international committee that figures out, or that makes some sort of standards to actually read the amino acids, read the nucleotide sequences, read the DNA sequences, read the RNA sequences. And that sort of standards are made by IUPAC, and they also make the alphabets. For example, they will decide if a DNA is ambiguous, if it contains N, it is considered as ambiguous or not, that sort of scenario. So BioPython allows us to utilize those sort of issues into and make a better decision about our sequences. For example, all you need to do is you need to import the alphabet module from the alphabet class of BioPython package. And there are other alphabets also available. For example, generic DNA. We will discuss that in the next video, and this is basically just an introduction of the alphabet. So how can utilize alphabet? So first of all, I have actually declared a sequence, a code basically that allows us to read a sequence from the user. So let's not waste our time on that. Let's just directly input the sequence. And okay, so basically it will then print out this is basically a string variable, and after that it will be converted into a sequence object. And then we will print its type, which will be sequence type. And over here we are doing a different thing. That will basically it is asking the alphabet class that, okay, this is my sequence object, and please tell me its alphabet. What sort of alphabet is it? Is it DNA? Is it RNA? Is it protein? Whatever it is, it will tell. But that is only possible if we have already defined the sequence alphabet in our declaration of the sequence object over here. We needed to define it over here. So now it will not tell us anything that it is either is it a protein, either is it a RNA, either is it DNA. So once the program runs, and now you can see that it is just displaying simple as alphabet, that okay, I don't know what sort of sequence object that you provided, but we can change that. We can obviously change that. And so let's go back and let's copy the entire code. And now we need to also import a different module from our class that will decide. Let's just basically, you don't need to worry about this, I will explain this in the next video. And